Assalamualaikum warahmatullahi wabarakatuh. Today we'll be presenting an article entitled Salmonella enteritidis, RFBD enhances bacterial colonizations and virulence through inhibiting autophagy. The significant discoveries in this article are the RFBD gene from Salmonella enterica, Cerebra enteritidis SE enhances bacterial colonizations both in vivo and in vitro by influencing autophagy, which is a cell cleaning process. It increases the inhibitions of autophagic flux, thereby reducing both the virulence and bacterial load. This offers fresh insights into combating infectious diseases by manipulating autophagy, potentially leading to more effective treatments. The hypothesis suggests that the RFBD gene plays a crucial role in manipulating autophagy within host cells. Next, the objective of this study is to investigate how RFBD gene from Salmonella enteritidis inhibits host autophagy and its effect. Additionally, to assess how RFBD-mediated autophagy modulations affects bacterial colonizations and virulence both in vivo and in vitro. Moving on to the methods. The first method used analysis of Salmonella infections in liver and sacum of female C570L/6. This analysis aimed to measure LC3-2 levels in the liver and sacum of mice and to evaluate how rapamycins and 3-MA affects autophagy and the survival of infected mice. The following method employ Western blotting to assess antibody expressions levels in infected HeLa cells. Finally, autophagosome analysis was performed on HeLa cells to quantify the number of L3 puncta using a high content analysis.
Thank you, Azhani. First, let's define the key terms. Z11 is the original Salmonella strain with the RFBD gene, while Z11 delta refers to the variant where the RFBD gene has been deleted. The double colon strain has the RFBD gene reintroduced. In vivo method shows that the absence of the RFBD gene in Z11 delta leads to the higher levels of LC3-2, a marker of autophagy, in the liver and cecum of infected mice. This suggests that the RFBD gene helps Salmonella survive better by reducing autophagy in the host cells. For in vitro method, rapamycin, an autophagy inducer, binds to FKBP12 protein in Salmonella, interfering with mTOR signaling and stimulating autophagy. Conversely, 3-methyladenine, an autophagy inhibitor, blocks the initiation of autophagosome formation. Western blot analysis reveals that the absence of RFBD gene leads to increased expression of ULK1 and BACLIN1, key autophagy-related proteins, while decreasing the levels of P62, which helps in cargo degradation. Finally, the autophagosome analysis showed that Salmonella infection, regardless of the presence or absence of the RFBD gene, can induce autophagy. However, the abnormal positioning of autophagosomes near the nucleus membrane suggests that Salmonella may interfere with the proper function of the autophagic process. Now, I will pass the next presentation to Izzah.
Thank you, Husna. Next, I will talk about the conclusions. Results indicate that RFBD-mediated autophagy helps Salmonella to escape the immune system. Through autophagy inhibition, RFBD has facilitated bacterial colonization both in vitro and in vivo. Do we agree with the conclusions of the authors? Overall, it can be concluded that every aspect is satisfactory, but there are still unanswered questions where the precise regulatory mechanisms that plays a crucial role in preventing Salmonella from autophagy is still under study. Did author consider either conflict or supporting evidence? It can be said that results of each discussions are accompanied by supporting evidence from various reliable and credible reference sources. For example, to illustrate the importance of role autophagy modulation in the pathogen's clearance, the author analyzed comparison using external reference and turned them into the comparative conflicts by examining several of different species like Salmonella, Shigella, Staphylococcus aureus, and also Listeria monocytogenes. Limitations of this research. First, there is less reliance on completeness or significant discovery in cases where specific mood rules needs for further investigation. Second, this paper does not offer any other methods for conducting future research, such as offering specific suggestion for prospective enhancement or direction for additional study. It will be ideal to show the public to take the proactive stance by supporting more research that fill in gaps found and broaden the knowledge base. Thank you for your attention. I hope you found this presentation insightful.